In this lecture, we are going to cover the digestive system, or you can refer to it as gastrointestinal physiology. So let's review the anatomy of the digestive organs before we get into the physiology. The digestive system is made out of the organs of the alimentary canal. The alimentary canal refers to all the organs that food physically passes through. So your digestive tract, or your gastrointestinal tract, is technically a tube that is open at two ends. And it's just the tube kind of coils up into the different organs, such as the mouth, where the food enters. Food then passes into the pharynx, which is basically your throat when you swallow, and down the esophagus, which travels through the thoracic cavity. And when the esophagus travels through the diaphragm, and you enter into the stomach. And then the tube continues from the stomach. You have your coiled small intestines, and remember you have three regions of the small intestines. The first part is the duodenum, then you have your jejunum, followed by the ileum at the distal end, and then the ileum feeds into your large intestines. And the large intestines travel kind of up and around the small intestines. So it travels up the right side of your body. That's your ascending colon, transverse colon that travels across the abdominal cavity, descending colon that travels down on the left side, and then you have your sigmoid colon where it makes that S-shaped, followed by the rectum. Your digestive system, when food is in the digestive tract, it's considered to be outside your body. I know that sounds a little odd, but because the tube is open at both ends, it's not open to the internal cavities of your body. Now there's other organs that make up the digestive system, and those organs we consider accessory organs. So accessory organs are other organs that play a role in digestion, but they're not part of the alimentary canal where food passes through. So you have your salivary glands, which produce saliva that assists in digestion. Remember, you learned in anatomy the different salivary glands you have. You have your parotid, which are up here in front of your ear, kind of by the masseter muscle. You have the submandibular glands and then the sublingual glands, which are under the tongue. Teeth can be considered an accessory organ that plays a role in chewing, assists in digestion. You have your pancreas, which sits here behind the stomach, and the pancreas produces digestive enzymes that help with digestion. You have your liver. The liver's role it plays in digestion is that it produces bile, and bile gets stored in the gallbladder, and that bile is going to be dumped into the small intestines to assist with fat digestion. So those are considered accessory organs of digestion. Okay. The other anatomy we need to review is the histology of the alimentary canal, so that when we look at the physiology, you'll be able to picture where these reactions and absorption and different things are taking place. So there's 4 layers if you look at a cross-section of the alimentary canal. For example, remember your small intestines. If you took a cross-section and you looked at the tissues, you have the mucosa, which is the innermost layer that lines the lumen, and that mucosa, that's going to be epithelium, okay, because it's on a surface. Then you have your submucosa, which lies underneath the mucosa, the epithelium, and that'd be this layer here, and that's going to be connective tissue, and it's usually areolar connective tissue. Then you have a layer of muscle called the muscularis externa, and there's 2 layers of muscle along the digestive tract. There's the inner circular layer, and then the outer longitudinal layer of smooth muscle. The only place where you actually have 3 layers of the muscularis externa is in the stomach. So the stomach has an inner circular and an outer longitudinal layer, and a 3rd layer that runs obliquely at an angle. And that gives the stomach the ability to churn, whereas your other digestive organs are basically moving things along the tract. And then the outermost layer would be the serosa. Okay, that's going to be epithelium. It's basically a serous membrane because it's actually it's found in a cavity, the abdominal cavity. And in the abdominal cavity, that serosa is called the peritoneum. The only place along the digestive tract where you don't necessarily have serosa is lining the mouth and esophagus, because they're not actually in a cavity. They'll have connective tissue around it. Alright, so let's jump into the physiology now that we've reviewed the anatomy. The basic functions of the digestive system. Most students can relate to the digestive system pretty well. It's a system you're pretty familiar with. So obviously, one function is to take food in, and that's called ingestion. So you take food in through the mouth. And then the food needs to move along the digestive tract so it can be processed. So another function is propulsion, moving the food. And that's done by the contraction of the smooth muscle that's along the tract. So there's two types of contractions that can occur to move food. There's what we call peristalsis and segmentation. So peristalsis is when you move food forward through the digestive tract. And this is involuntary waves of contraction of the smooth muscle and relaxation. The smooth muscle at one end of the digestive tract will start to contract, and it's like a wave of contraction followed by relaxation, and it moves the food forward. Segmentation is a slightly different type of contraction. If you think of the digestive tract as a tube, it kind of alternates its contractions in different areas of the tube. So one area will contract and then relax, and then a different area will contract and relax. And the purpose of segmentation is not necessarily to move things forward, but it's more to break the we call the food a bolus once it gets compacted, to break the bolus into smaller pieces or to mix the food with digestive enzymes and secretions. An analogy that I use to distinguish between peristalsis and segmentation is if you had a tube of toothpaste, and if you squeezed from the bottom of the tube of toothpaste towards the top to get the toothpaste out, that would be like peristalsis. Okay, you're moving the contraction up and in one direction. Segmentation would be like you squeeze anywhere you want on the toothpaste. So you just squeeze randomly in different spots, and the toothpaste would not necessarily move completely forward. It would kind of move back and forth and get mixed up a little bit. So we all know some people like to squeeze the toothpaste from the bottom, whereas other people will just come and squeeze it wherever they want. Alright, the digestive system obviously is to digest, and the definition of digestion is to break down food into smaller molecules. Now there's two different ways you can break food down. You can mechanically change the food, or then we call that mechanical digestion, or you could chemically change the food, and that's called chemical digestion. The difference is, in mechanical digestion, you're changing the physical structure of the food. So what we mean by that is, say you had a solid piece of bread you take in, and you start chewing it. When you chew it, you're breaking that piece of bread into smaller pieces. That would be mechanical because you're not chemically changing the structure. You're taking the bread and breaking it into smaller pieces. And that bread's made out of, for the most part, complex carbohydrates or starches. So when you break it into smaller pieces, it's still made out of starch, it's just smaller pieces. Okay, that'd be mechanical. Another example is if you took the solid piece of bread and you mixed it with digestive secretions to make it from a solid to a liquid. That'd be a physical change, and that would be mechanical. So areas where you have mechanical digestion would be chewing in your mouth, even mixing the food in the stomach would be mechanical digestion. Segmentation in the small intestines where you're mixing the food, that would be mechanical. Now chemical is you actually change the chemical structure. So chemical digestion is always going to involve digestive enzymes because it's going to facilitate a chemical reaction. For example, you have starches in your bread where you're going to have an enzyme called amylase that's going to break those starches, those polysaccharides, down into disaccharides and eventually monosaccharides. So you're technically breaking chemical bonds, and that would be chemical digestion. So anytime you're involving a digestive enzyme, you're going to have chemical digestion. The majority of your chemical digestion happens in the small intestines, but we'll learn some also happens in the mouth and stomach. All the food you take in, they're macromolecules, right? You take in polysaccharides or triglycerides proteins. Remember back in chemistry we learned about that these are large molecules. They're made up of monomers, like a protein is made up of many amino acids strung together. A polysaccharide is made up of numerous simple sugars strung together. Those macromolecules are too big for your cells to transport. Okay, and so they actually want them to be broken down into the monomers. So that's the purpose of your digestive system. It's going to break the proteins down into amino acids so that you can absorb the amino acids. It's going to break down the polysaccharides into simple sugars so you can absorb the simple sugars. So once the digestion happens and you break it down into those monomers, now your body has to absorb it from the lumen of the intestines and get it into the blood or lymph system so it can travel to the cells that are going to use it. So absorption is the transport of the nutrients from the lumen into the blood. Fats are also carried in the lymph vessels, so some gets absorbed into the lymph. And then whatever your body doesn't absorb, you need to get rid of. It's waste. So anything remaining in the digestive tract that isn't absorbed in the small intestines has to be eliminated from the body. And that's called defecation. For example, we can't cellulose, which makes up the cell walls, so that gets left in the digestive tract. Even some bacteria and different things will be left. Alright, so what regulates digestive tract activity, or the gastrointestinal tract? We're going to be looking at two things here. Activity of the digestive tract, the two things that it's doing mostly is it's moving things along, so that's involving contraction of smooth muscle. The other thing that's happening is there's a lot of secretions that get put into the digestive tract to aid with chemical digestion. We're talking about mostly through this lecture what controls the contraction of the smooth muscle and what controls glandular secretions. Okay, so keep that in mind. So there's a few things that regulate those two processes. And I'll list them here and then we'll go into detail about them throughout our lecture. So one is autonomous smooth muscle. Remember when we learned about smooth muscle, it had those pacemaker cells that are able to spontaneously depolarize and initiate contraction. So smooth muscle can contract without extrinsic controls of the nervous system. Or it can contract with extrinsic nerves controlled by the central nervous system. The other thing is there's a lot of receptors along the digestive tract that trigger these digestive reflexes. Your digestive system is monitoring stretch when organs fill, for example your stomach, osmolarity, so the chemical composition of the food or the chyme, the solute level, the presence of certain substrates, for example protein versus amino acids, so we'll talk about that. Also you learned a lot of hormones in the endocrine system. Well, guess what? You're going to learn some new ones today that you didn't learn about back then that play a specific role in digestion. So we're going to learn some of the gastrointestinal hormones that regulate smooth muscle contraction and glandular secretions. Extrinsic nervous system. So the extrinsic nervous system that controls the digestive system, glands and smooth muscle, would be what division? Smooth muscle and glands are controlled by the autonomic nervous system. Parasympathetic and sympathetic. And then something that's very unique and kind of makes digestive physiology a little complicated is the digestive tract has its own intrinsic nervous system. So it's a local control, and we'll talk about that. So let's first look at autonomous smooth muscle. We've learned that visceral smooth muscle is autonomous. That means that it can display rhythmic, spontaneous action potentials, and that can trigger the smooth muscle to contract. And remember, smooth muscle is connected by gap junctions. So you have these pacemaker cells that have what we call slow wave potentials that fluctuate towards threshold and away from threshold. Now, if they reach threshold, they're going to trigger a series of action potentials that will move through the tissue and cause that smooth muscle to contract. It will carry out these contractions so many times per day. However, it still can become more active or less active by extrinsic controls and other intrinsic controls. So for example, hormones, which would be an extrinsic control, can make the smooth muscle more or less active. make it contract more often or less often. Same with mechanical stress, such as stretch. Nerve stimuli, for example, from the parasympathetic nervous system, they can make that tissue more active. And the way that it does that is hormones in the extrinsic nervous system, for example, your parasympathetic nervous system dumping ACH onto the tissue, can cause these slow wave potentials to fluctuate closer or further away from threshold. So if they're fluctuating closer to threshold because of the presence of a hormone that makes the tissue more active, then you're going to have action potentials more frequently. And so what this looks like is you can have graded potentials in your smooth muscle. So you're looking at the action potentials up here. Down here you're looking at muscle contraction. So your vertical is force versus time. Up here you're looking at electrical activity in the tissue. So it's millivolts over time. So if you don't reach threshold, you're not going to have a muscle contraction. If you reach threshold and you fire one action potential, then you might have a muscle twitch. The smooth muscle contracts and then relaxes. But you can have summation in the smooth muscle. So if you have multiple action potentials, because you're fluctuating closer to threshold, then your muscle contraction of the smooth muscle is going to get stronger and last longer. So the force and duration of the muscle contraction is directly related to the amplitude and frequency of the action potentials in the tissue. Sometimes your digestive tract is really active, sometimes it's less active. It's mostly active when food is entered into it. Okay, it's less active when food is not there. So we're going to learn about that, how that stimulus causes the tissue to become more active. Why would the tissue become more active or less active? Well, a lot of it is its receptors detecting changes in the digestive tract. And when you detect those changes, it triggers a response. And so some of the receptors that you have in your digestive tract are mechanoreceptors, so they respond to stretch or pressure. So putting food into your mouth, you have pressure receptors that detect it, and it triggers saliva to be produced. Okay, we'll talk a lot about these reflexes as we go, but right now I'm just giving you an example. Or stretch, when food enters into the stomach, you have stretch receptors that detect that. You also have osmoreceptors, and they're detecting changes in osmolarity. Now remember, osmolarity is the level of solutes in a solution. When you eat something, you're increasing the osmolarity of, for example, what's in the lumen of your stomach, because now you have sugars, proteins, different things that are now in there, so you have a high osmolarity. Your digestive tract knows that there's food there and digestion needs to start taking place. And then also chemoreceptors. You have pH receptors, presence of certain substrates. You have some substrates that literally detect amino acid levels or the end products of digestion. How this works is your digestive tract actually knows how much digestion's actually occurred because it's a chemical reaction, right? If I have a polysaccharide and I need to get to the simple sugars, your digestive tract knows kind of how far along you are because it can detect the presence of the substrate, which is a polysaccharide, in proportion to the amount of the end products you have, sugars. So if you have a lot of polysaccharides, very few sugars, you have a ways to go with digestion before you are done. If you have a lot of sugars, very little polysaccharides, your digestive tract knows, okay, we're done breaking this down, now we can start absorption. So these types of receptors will detect changes or stimuluses in the digestive tract, and it will trigger a reflex, okay, or a feedback loop that will either activate or inhibit digestive glands and activate or inhibit smooth muscle. So it will either make the digestive tract more active or less active. Now I refer to these as short or long reflexes because remember, almost all of our organ systems have extrinsic controls that come from outside the organ. So that would be like your nervous system or hormones. And then there's local controls, or what we would call short reflexes, where the stimulus is detected in that organ and you have the response in that organ. So let's compare what a long reflex is versus a short reflex. Okay. Long reflexes, what that means is the stimulus may be within or outside the gastrointestinal tract. So what I mean by that is you can alter the activity of your stomach just by thinking of food. So the thought of food is the stimulus. Well, that's occurring outside the stomach. That's happening in the cortex of your brain. And then just the thought of it can trigger your autonomic nervous system to send parasympathetic impulses to the stomach and cause your stomach to start to contract and churn. That would be an example of a long reflex because the stimulus is outside of the organ that becomes your effector. So this involves the integration in the central nervous system. It's going to have to involve your central nervous system. Digestion is mostly controlled in the medulla, and then the output would be sent via autonomic nerves to the gastrointestinal tract. In general, your parasympathetic nervous system will make your digestive tract more active because it would make the smooth muscle and the glands more active, whereas sympathetic makes digestion less active. Okay, so I kind of covered this. Extrinsic controls can alter the muscle activity or the gland activity. It can also trigger certain hormones to be secreted. It can modify intrinsic activity, which we'll talk about next. Long reflexes are always related to when you coordinate different parts of the digestive system. Do you remember back in our first lecture, we talked about feed-forward mechanisms? And that's where a response happens in anticipation of something occurring. Well, your digestive tract is filled with these feed-forward mechanisms. And the idea of your digestive system is if food is in my mouth, the next place it's going is down the esophagus and to my stomach. And if I know that food's in my stomach, I know it's going to go eventually to my small intestines and large intestines. So what happens is when you just chew food, it triggers the stomach to start making gastric secretions. It's a feed-forward mechanism because your stomach's getting ready in anticipation to the food coming there next. Or you've had this happen where you could smell and think of food and your salivary glands start to produce saliva. That's a feed-forward mechanism because you're assuming that if you can smell the food and see it, it's coming to your mouth next. That's not always the case, but that's what your body thinks. So here's a little picture. This might help a little better. Okay, you can have an external stimulus, such as sight and smell of food. It gets detected by sensory receptors, maybe your photoreceptors, and it's going to go to the brain. Most of it's getting controlled for digestion in the medulla. And that will trigger your autonomic nervous system neurons, sympathetic and parasympathetic, to stimulate neurons that are found in the muscularis externa. You might have learned this in anatomy, but it's kind of a minor detail you might have forgotten. There's some plexuses, which are nerve branches that are found in the layers of your digestive system, and the myenteric and submucosal plexes are influenced by your sympathetic nerve and parasympathetic nervous system. And that will trigger the smooth muscles or the glandular cells to either contract or relax or produce more secretions, or endocrine glands to produce hormones. So this is a long reflex. Another thing that can happen with long reflexes is it could be that you detect something within the organ. So it could be a local stimulus, such as a change in pH, and it gets sent to the brain and then out the autonomic nervous system. That's also considered a long reflex. A short reflex, though, is digestive system has its own intrinsic nervous system, and it's called this enteric nervous system, and that involves the myenteric and submucosal plexuses. The sensory gets detected, or the stimulus gets detected in the digestive organ, and those neurons then trigger smooth muscle and the glands. So it doesn't actually have to go to the central nervous system. This would be considered a short reflex. So it involves the enteric nervous system, which is a little mini nervous system inside the digestive tract. So the entire reflex arc is carried out within that tract, and it responds to a local stimulus. So that means that the stimulus is there, the receptor, the integration center, the output, and the response all happens within that organ. So it's going to be coordinating local activity, and it can work independently of the central nervous system and your extrinsic nerves, or it can be influenced by your extrinsic nervous system. Now the other thing besides your extrinsic nervous system, which is your autonomic nervous system, and your enteric nervous system that's within the digestive tract, you also have hormones that can regulate the activity of the muscle and glands. So there's numerous gastrointestinal peptides that act either as a hormone, because they'll travel in the blood to a nearby digestive organ, or it could be a paracrine signal where the peptide gets released and it just affects local cells, not actually traveling in the blood. They can also travel in the blood to the brain, and their levels can either cause you to be hungry or full. That's satiety. Alright, so we just talked about the different things that influence digestive activity. And the main thing we're looking at is smooth muscle contractions and glandular secretions. So when I say that the digestive tract becomes more active, that means the smooth muscle contracting more often and you're producing more secretions. If it becomes less active or it's inhibited, smooth muscle contracts less often and you're producing less secretions. So now what we're going to do is we're going to kind of move through the digestive tract, taking the same path that food would take, and we're going to look at the functions of each of these digestive organs. So we'll start with the mouth. So that's where we chew our food, but this is the beginning of mechanical digestion because you're breaking the food into smaller pieces. Another physical change that's happening is you're taking the food and mixing it with saliva that your salivary glands secrete into your mouth. And that saliva has mucus that allows the food to stick together, and you form this compact ball of food that we call a bolus. The other thing that's found in saliva is the enzyme amylase. And amylase will start your chemical digestion, and this is of carbohydrates. So amylase is the enzyme that takes the substrate polysaccharides and breaks it down into, mostly we say disaccharides, but the most common disaccharide that it breaks it down into is maltose. So your chemical digestion of carbohydrates begins in the mouth, and you're also beginning your mechanical digestion of all food. So let's look at saliva. Remember, you have your parotid gland, your submandibular, and your sublingual that have ducts that empty the saliva into your mouth. These salivary glands have two types of cells. They have serous cells that produce a watery secretion that has a lot of enzymes in it. And then your mucus cells produce the mucus component of the saliva. So saliva is mostly water. It has the digestive enzyme amylase. And then the mucus cells make the mucus, and the purpose of the mucus is to kind of moisten the food, but also mucus is sticky, so it helps hold the food together so you can form a compact ball called a bolus to swallow. And the other thing you'll find in your saliva is some defense mechanisms, because taking in food is one way you take in pathogens from the outside. And so you have to have some body defenses. And so there's an enzyme called lysozyme that's in saliva that has an antibacterial action, will destroy bacterial cells. And IgA is a type of antibody that you'll find in your salivary secretions. So the reflex that's involved in producing saliva, there's two types of reflexes. Okay? There's what we call a simple reflex and then the acquired reflex. Another name for the acquired reflex is conditioned because it's learned. The simple reflex you're born with. So the simple reflex is when you take food in, there's pressure receptors in your mouth that detect that the food is now in your mouth, and then there's also chemoreceptors that detect the substrates like carbohydrates and proteins and so forth that are in your mouth. So those get detected, and then it's sent through a sensory neuron to the salivary center, which is in the medulla, and then the output is sent through the autonomic nervous system to the salivary glands, which would be your effector, and the response would be to increase saliva secretion. So this is the simple reflex. It's the actual physical food getting put into your mouth that triggers the saliva production. The acquired reflex is learned. It involves your cerebral cortex. This is Pavlov's dog, where he taught the dog to associate the ringing of the bell with food. Okay, so when you'd ring the bell, the dog would start salivating. When you think of food or you smell food or anything, you become aware that the food's there, so that's the cerebral cortex, and that's enough to trigger increased saliva production. Okay, so that's the conditioned or the acquired reflex that's learned. Now, who controls the saliva or the salivation? It's the autonomic nervous system, or the ANS. Whenever I teach this to non-majors, or I'm trying to simplify who controls production of saliva, we usually just say that parasympathetic nervous system always increase digestive secretions. Because remember, parasympathetic nervous system is nicknamed rest and digest, right? The parasympathetic nervous system is responsible for producing a larger volume of saliva. So when parasympathetic is dominant, you produce a larger volume of saliva. It's more watery and it's rich with amylase. It doesn't mean that your sympathetic nervous system, though, doesn't influence saliva production, because it does. But what happens is when the sympathetic nervous system is dominant, you're producing a smaller volume of saliva and the mucus cells are more active. And so you're producing more mucus. So you're producing more of a really sticky mucus, and that's why when you're in sympathetic or you're nervous about giving a speech, your mouth feels all dry and pasty. It's because you're producing a less watery volume and it's more sticky. So technically both parasympathetic and sympathetic play a role in producing saliva, but the response is different. Parasympathetic increases the secretion, sympathetic would decrease. So once you packed your food into a nice bolus, you now need to swallow it, and that's called deglutition, or swallowing. And this is also a reflex. Part of it is voluntary, part of this reflex is involuntary. So what that means is you initiate it, but after a point it becomes involuntary and you can't stop it from happening. We divide it into two phases, the buccal phase and the pharyngeal esophageal phase. Some people will divide this into three separate phases. But the buccal phase is when the bolus is forced into the oropharynx. So you use your tongue and your soft palate to initiate swallowing, and the food gets put into the pharynx, the oropharynx. But once it gets put back there and starts to move down lower into the laryngopharynx, it becomes involuntary, and the smooth muscle contractions take over. So the pharyngeal-esophageal phase is controlled by the medulla and the lower pons. Food will move into the laryngopharynx, and through peristalsis, it'll move down the esophagus. So remember, by like a wave-like contraction of the smooth muscle, it'll move down the esophagus until it gets to the end of the esophagus, which is basically where the diaphragm divides the esophagus from the stomach. And there's a little sphincter there, and then food will move through that sphincter into the stomach. And so then food enters into the stomach. There's different names for this little sphincter that is here between the esophagus and the stomach. I learned it as the cardioesophageal sphincter because this is considered the cardiac region of your stomach, and your heart is right above here. Or some books refer to it as the gastroesophageal sphincter. I looked up in your book and it actually calls it just the lower esophageal sphincter. So there's a lot of names for it. I'm not going to necessarily quiz you on that. You just need to know that there's a sphincter there dividing the esophagus and the stomach. Okay. And what can happen is the diaphragm plays a role in kind of keeping the tension on that sphincter. And so if anatomically your diaphragm moves up from there, you can have what we call acid reflux, where some of the acid moves into the esophagus because you don't have that tension there from the diaphragm. So sometimes anatomically people will have that. Other people sometimes have a herniated stomach that herniates up in through the diaphragm and into the thoracic cavity, and they could have acid reflux or what we call heartburn. And that can be dangerous because it can lead to ulcers in the esophagus that can then bleed and cause problems. So now we're into the stomach. What happens with food in the stomach? The main function of the stomach is to actually mix and store the food until it can be emptied into the small intestines. Very little actual digestion occurs in the stomach, and hardly any absorption occurs in the stomach. It's basically a storage place. It will take the bolus and change it both physically and chemically. The physical or mechanical digestion that occurs is when you mix the food with the gastric juices. So you're going from a solid bolus, mixing it with a bunch of gastric secretions, which we'll learn about the different secretions that get secreted into your stomach, but it's turning that bolus into a liquid, and we call that liquid chyme. So that would be a physical change. There is some enzymes that start the digestion of proteins. So you will start chemical digestion of proteins with the enzyme pepsin in the stomach. For the most part, carbohydrate digestion gets put on hold in the stomach because the enzyme amylase can't work at that high acidity in your stomach, and then the digestion of carbohydrate will continue once it enters into the small intestines. So basically you just have protein digestion happening here. You're not really absorbing any nutrients in the stomach. They've seen that some medications or drugs can be absorbed in the stomach, such as a small amount of alcohol. They say, I think it's aspirin that can maybe be absorbed a little bit from the stomach, but for the most part, no absorption occurring. One other function of the stomach is the secretion of a secretion called intrinsic factor. An intrinsic factor is required for the absorption of vitamin B12 later on in your small intestine. So we'll talk more about that. Well, as you get older, you start to secrete less intrinsic factor, and so you might have trouble with vitamin B12 absorption, or some people just genetically aren't producing enough intrinsic factor, and they have to take intrinsic factor so that they can absorb vitamin B12. So those are the functions. It mixes and stores. You do have mechanical digestion and some chemical digestion, and then it plays a role in the absorption of vitamin B12 because it produces the intrinsic factor. There is long and short reflexes in the stomach. It's kind of the same thing. Just the thought of food can get your stomach activity going, so that would be a long reflex that would increase secretion and motility in the stomach. Or there can be little local reflexes that make the stomach more active. So just food entering into the stomach, you have receptors that will detect the substrates like amino acids or the acidity and the osmolarity. Through a local reflex or intrinsic reflex, you would affect the motility and secretion of the stomach. So now let's look at how digestion occurs in the stomach. We'll first look at gastric motility, how the stomach contracts and moves food through the stomach. There's 4 phases of gastric motility that we'll work through. First is gastric filling, then there's storage, mixing, and emptying of the stomach. We will go through each of these phases in our next few slides. So let's start with gastric filling. This is pretty easy. Basically, you're filling the stomach up with food that you've swallowed that's traveled down the esophagus. And as the stomach fills, it will actually relax, and the rugae, remember the folds that are within the stomach, will start to flatten out. The stomach can actually dilate and hold quite a bit of food. It can fill to about 1 liter, which is quite a bit. Now when you look at the areas of the stomach, remember you have your fundus up here, the body, and then the pyloric region. And then there's a region between the body and where the pyloric sphincter is called the antrum. And this area is very muscular, whereas up towards the top, it's not as muscular and the contractions aren't as strong. So you have peristaltic contractions that begin up at the body and travel towards the pyloric sphincter. Well, the contractions up here at the body are a little weaker, and so little mixing occurs up here. So we say when there's food or chyme up here in the body area, we would consider that gastric storage. You're storing it there until you can mix it and then empty it. So where most of the mixing occurs is in the muscular area of the stomach called the antrum, which is about right here. So you have really strong peristaltic contractions in this area, and it will push the chyme towards the pyloric sphincter, and then it kind of bounces back against that closed sphincter, and you get a mixing action. Now what you're mixing your food with in the stomach is a number of gastric secretions. If you remember the histology of your stomach, there was gastric pits. So if this is the lumen of your stomach up here, and this is the rugae here and here. It's lined with epithelial cells, simple columnar, and then you have these gastric pits that extend deep into the submucosal layer. Well, in these pits, you have cells that produce the gastric secretions that will be secreted through the duct and then onto the luminal surface of the stomach to be mixed with the food. Different things make up the gastric secretions. You have some exocrine secretions, which means it gets secreted into the duct and onto a surface. And here in the neck area and lining the lumen of the stomach, you have what we call mucus cells. And this is easy, they produce mucus. The purpose of the mucus is it produces a layer that protects the wall of the stomach from harsh acids. The harsh acids and enzymes that get secreted into your stomach can actually digest the lining of your stomach, so that mucus creates a barrier. They have found that some things compromise this mucus barrier, such as the bacteria H. pylori. And H. pylori, its metabolism affects that mucus barrier, and then what can happen is the acids of the stomach can then kind of eat away at this epithelium and into the connective tissue, and this is called an ulcer. It can be really dangerous if you get down to where the blood vessels are in this connective tissue, and you can end up with a bleeding ulcer. Another secretion is produced by cells called parietal cells that are along these gastric pits. Parietal cells produce the acid, HCl, and acid plays a role in activating some of the enzymes, and also it denatures some of the proteins, and it will also kill any pathogens that may have gotten in through your food. Another secretion that comes from the parietal cells is what we call intrinsic factor. But intrinsic factor plays a role in the absorption of vitamin B12 in your digestive tract. And then there are chief cells, and chief cells produce pepsinogen. Now pepsinogen, remember we learned GEN on the end means that it's inactive, and when it gets activated, it's ion. You've already learned about angiotensinogen, gets activated into angiotensin. You learned about fibrinogen, gets activated into fibrin. Okay, so you've learned some of these enzymes. Pepsinogen is its inactive form and will get secreted into the lumen, where then it will be activated into pepsin. The reason you secrete it in its inactive form is because it degrades proteins, and your cells and tissues are made out of proteins. So you don't want it to get activated until it's up here in the lumen where you have a mucous membrane or a mucus layer to protect the underlying cells. If it's active down here when it gets secreted, it might start degrading your own tissues. So you can see here how it gets activated. It's actually the HCl, the acid, that activates the pepsinogen into pepsin. And then pepsin will break peptide bonds between amino acids of your protein to form smaller peptide fragments. The chemical digestion of proteins begins in the stomach. Now, you also have some endocrine cells in the gastric pits called G cells, which produce the hormone called gastrin. What gastrin does is it stimulates gastric acid secretion to occur more. Gastrin actually activates the parietal cells to produce more HCl. Then you have some paracrine chemicals that get released. Remember, paracrine signals work on nearby cells. So there is some cells called HCl cells that secrete histamine. And histamine also stimulates the secretion from the parietal cells. So now you have both gastrin and histamine, which will increase the hydrochloric acid production from the parietal cells. Now you also need to be able to shut off these gastric secretions. You don't want to just continually produce them. So there's a negative feedback loop, which involves cells called D cells. And D cells produce what we call somatostatin, which is a paracrine. And somatostatin will then go and inhibit the secretion from both the parietal cells, the ECL cells, and the G cells. It basically shuts down gastric secretions. Now what triggers the secretion of somatostatin is a really low pH. When the pH gets too acidic, you want to shut off HCl production. So that's what triggers the release of somatostatin. So how do we regulate gastric secretions? How do we produce more gastric secretions? How do we produce less gastric secretions? Well, it's going to be controlled by both neural and hormonal mechanisms. And there's things that will cause more gastric secretions to be produced, so we'd call that excitatory or stimulatory events. And then there's some things that happen that actually inhibit gastric secretions, which we would call inhibitory events. And there's 3 phases that regulate gastric secretion. There's what we call the cephalic phase, which these are things that happen prior to food entering the stomach to either excite or inhibit secretions. Then there's the gastric phase, and these are events that happen to cause gastric secretions to increase or decrease once food enters the stomach. And then there's the intestinal phase, which are things that excite or inhibit gastric secretions once the food enters into the duodenum. So what we're going to do is we're going to go through each of these 3 phases, and we're going to talk about what excitatory and inhibitory events occur before the food gets to the stomach, what events occur when it gets to the stomach, and the events that occur once it leaves the stomach and enters the intestines. So let's first look at the cephalic phase, prior to food entering. Cephalic refers to the head region anatomically, so I think of it as these are things that happen within your head. So things that will excite gastric secretions. If you just think of food or see food, that will trigger a feed-forward mechanism that increases gastric secretions in the stomach. The other thing is just putting food in your mouth. The taste receptors, if you smell food, and then when you put food in your mouth, there's pressure receptors that detect that also. So putting food in your mouth will trigger a feed-forward mechanism and increase your gastric secretions. The other thing is your parasympathetic nervous system. So remember, the parasympathetic nervous system is your rest and digest. So whenever your parasympathetic is dominant, you're going to be increasing all your digestive secretions and motility. So the vagus nerve is your big parasympathetic nerve that travels to your visceral organs, and he would release ACH because you remember that parasympathetic postganglionic neurons release ACH. So the neurotransmitter ACH can activate parietal G cells and ECL cells to produce more secretions. Some events that can happen in your head that will actually inhibit your gastric secretions are kind of psychological things, such as if there's even food that you don't like, can cause you to lose your appetite, or depression. The other thing that will inhibit gastric secretions is if there's a decrease in parasympathetic activity. So, for example, if you're in sympathetic, then gastric secretions would be inhibited. Now, once food enters the stomach, things that will excite gastric secretions is the actual distension of the stomach. So as the stomach fills, stretch receptors are triggered and gastric secretion will increase. The other thing is certain substrates being in the stomach. For example, chemoreceptors that detect peptides or proteins, caffeine, and then also alkaline pH. So if the pH is alkaline, it will trigger the release of gastrin into the blood. And remember, gastrin then increases secretions from the parietal cells and the ECL cells to produce more hydrochloric acid. Now, the only thing that's going to inhibit gastric secretions once food enters the stomach is if this acidity gets too low. So if your pH gets really low and very acidic, you're going to want to inhibit hydrochloric acid secretion. So if the pH gets lower than 2, that will trigger the release of somatostatin from the D cells. And remember, the somatostatin will then go and inhibit gastrin secretion and inhibit parietal cells and ECL cells. So this is kind of a good summary of these gastric secretions. So you can see here that, remember, when the D cells release somatostatin, what triggers them to be released is if the stomach gets too acidic, and that will inhibit gastric acid secretion. I won't go through the whole thing, but you can see like parietal cells, what they secrete, gastric acid and intrinsic factor, what will stimulate their release, acetylcholine, because that's from your parasympathetic nervous system, gastrin from the G cells, histamine from the ECL cells. Acetylcholine triggers the release of all these, so increased parasympathetic activity would increase gastric secretions. And then you can see the function of each secretion here. And this is more of just a picture visual of the information you just learned. So you have your different cells here: G cells that secrete gastrin, D cells secrete somatostatin, parietal cells secrete hydrochloric acid, chief cells secrete pepsinogen. And it's showing this negative feedback loop. So when certain substrates enter into the stomach, such as amino acids or peptides, that will get detected by a receptor in the stomach, and through the enteric nervous system, that would trigger the G cells to release gastrin. But gastrin can also be triggered by parasympathetic activity, the vagus nerve, releasing ACH. And you can see that the vagus nerve would also stimulate the ECL cells, produce more histamine, which activates the parietal cells. And then the parietal cells can also be activated directly by ACH. And then you can kind of see here the negative feedback loop. So if it gets too acidic, too many hydrogen ions, that would activate the D cells to release somatostatin, and somatostatin shuts down the secretions, inhibits the secretions by inhibiting G cells, parietal cells, and chief cells. And this is a picture just looking at a single parietal cell. So you'd have the lumen of your stomach here. Here's a parietal cell, and then this would be the basement membrane of the cell. And you can see how it produces its hydrochloric acid and the things that trigger it. Okay, we talked about that. Gastrin, ACH from the parasympathetic nervous system, and histamine from the ECL cells would all activate more hydrogen ion secretions. And they do it through, guess what? Secondary messenger pathways, which you learned about. These two are through calcium secondary messenger pathways, and histamine works through a cyclic AMP pathway. You may have heard that caffeine can increase the acidity of your stomach. One, coffee is acidic, but the other thing is caffeine, the drug caffeine, will cause the cyclic AMP pathways to stay active longer, so that can increase hydrochloric acid secretion. And you can see that it's a hydrogen potassium antiport channel, and it's going to pump hydrogen ions into the stomach in exchange for potassium ions. And then chloride just follows the positive hydrogen ions. So on the basement membrane side, you actually have chloride bicarbonate antiporter, similar to the one you had in your red blood cells. Remember the chloride shift you learned about that happens in red blood cells? So here, if you're pumping chloride ions in, you got to swap it for another negative ion. So you swap it for a bicarbonate, and then the chloride will just follow the hydrogen ions into the lumen of the stomach. Now, something that's a little interesting is as you produce hydrochloric acid in the stomach, then you're producing more bicarbonate here on the basement membrane side. That bicarbonate is going to go into the blood. And so they call this the alkaline tide, because bicarbonate is a base, and the blood leaving the stomach is more basic than elsewhere in your body. It's called the alkaline tide. From there, it doesn't stay alkaline as it travels through your body, because usually it then goes towards the pancreas, and the pancreas will then use this bicarbonate from the alkaline tide to produce what we call sodium bicarbonate to neutralize the chyme that comes from the stomach. So it's kind of a little interesting mechanism you have there. Alright, now we're going to look at the intestinal phase. So this is what happens to gastric secretions once the chyme empties from the stomach and into the intestines. Now this phase is mostly inhibitory, because the intestines, he only wants to deal with a little bit of the chyme at a time. He wants a small volume. And the reason is your intestines want to maximize chemical digestion and absorption. And in order to do that, he only wants to work on a little bit of material at a time, so he can make sure that he breaks everything down and then absorbs it. If he gets too much, he can't be as efficient at that. So what he does is when the chyme gets into the intestines, into the duodenum, the first part, it will try to slow down the gastric secretions from the stomach. So what triggers this inhibitory phase is when there's distension of the duodenum, so just the chyme filling into the duodenum causes distension, and then the presence of fatty acidic. It's acidic because it's coming from the stomach, fatty because you haven't even started fat chemical digestion yet, or if it's hypertonic chyme. Hypertonic refers to the number of solutes in the chyme. Well, you haven't had any absorption happen yet, so the chyme's pretty hypertonic, a large amount of solutes. So what happens is when it enters into the duodenum, there's a reflex, a local reflex that's intrinsic, and then the vagus nerve, okay, the vagal nuclei that's in your medulla can trigger the closure of the pyloric sphincter. And then there's some hormones that are going to be released, and you have some endocrine cells in the lining of the small intestines. And two hormones that you're going to hear about throughout this lecture is cholecystokinin, or we abbreviate that CCK, and secretin. And they will go back and work on the stomach. They'll travel in the blood to the stomach, and they'll decrease gastric secretions and slow gastric motility. Okay, so they're going to slow the stomach down. Another hormone is gastric inhibitory peptide. Now, he was named because he inhibited the stomach. Okay, gastric inhibitory peptide. But then what they found just more recently, they found what this hormone does is it actually also works on the pancreas to promote insulin release. Now this is interesting because it's a feed-forward mechanism. When chyme enters into the duodenum, there's a lot of sugar in there that you need to absorb. And the small intestines is going to absorb the sugar and it's going to go into the blood. And you're going to need to have insulin in the blood to start getting the glucose out of the blood into the cells. So it's a little feed-forward mechanism that when the chyme enters, before you even start absorbing the sugar, it will trigger insulin to be released so that insulin's already in the blood ready to initiate the uptake of glucose to the cells. So this is just a summary of what we just talked about. Stimulatory events, things that will increase gastric secretions, or I call them excitatory events. Same thing, stimulatory, excitatory. And then events that will inhibit gastric secretions. And then this chart is divided into cephalic phase, gastric phase, intestinal phase. Now here in the intestinal phase, I mostly talked about just that the intestinal is mostly inhibitory. That's what you need to focus on. Here they're talking about something about how when it first enters, it stimulates it for a second, but don't worry about excitatory. Know that intestinal is mostly inhibitory. Alright, now back to gastric motility. We talked about filling, mixing, and now we're going to talk about how you empty it. You put all that gastric secretions, mix it around with the bolus, and once it gets nice and liquid-like, you're going to empty the stomach into the duodenum. So there's peristaltic waves that happen that move from the fundus towards the pyloric sphincter, and they'll empty a little bit of the chyme into the duodenum. Now the rate of gastric emptying is controlled by both gastric and duodenal factors. So things that happen in the stomach and things that happen in the duodenum. So what we're going to talk about is now what will speed up gastric emptying is the smooth muscle contraction of the stomach. How quickly you're carrying out these peristaltic contractions. So again, there's things that will increase gastric motility, which would increase emptying, and there's things that are going to decrease gastric motility and decrease your emptying rate. It takes about 4 hours, they say, for an average meal for your stomach to empty completely. So what's going to increase gastric emptying? What will increase gastric emptying is things that happen in the stomach, gastric factors. So the amount of chyme that enters into the stomach will empty at a rate that is proportional to the volume of chyme. So if you have a larger volume, your stomach will need to speed up its rate of emptying. If you have a smaller volume, it can empty at a slower rate. The idea here is you're changing the rate. You can still get your stomach to empty somewhere between 2 and 6 hours. So we average that to about 4 hours for your stomach to empty. Okay, even if you eat a lot, even if you eat less, it will speed up the rate because the rate is proportional. So this is triggered by the distension. So what's increasing the gastric motility? Well, there's distension. So when your stomach fills, it triggers smooth muscles to start contracting and increase the peristaltic waves. The other thing is the fluidity of the chyme. Your stomach doesn't want to empty until it's mixed the bolus with the gastric juices. So it's kind of able to detect how fluid the chyme is. So these are intrinsic controls, the stretch and the fluidity. Then there's some extrinsic controls, the vagus nerve. So parasympathetic activity would increase gastric motility, therefore increase gastric emptying. And then there's the hormone gastrin. And this is a little controversial. We just learned that gastrin would increase gastric secretions. There is some evidence that gastrin would also increase gastric motility. The last, most recent thing I read was a study where they were saying, like in animals, that they found that gastrin does increase motility along the digestive tract. Alright, things that are going to decrease gastric motility and therefore decrease gastric emptying is duodenal factors. Now, the duodenum wants to reduce peristaltic activity in the stomach until it's ready to accommodate more chyme. So things that will decrease gastric emptying. How fatty the chyme is that the duodenum is receiving. You haven't even started fat digestion yet. And fat digestion is slower, takes longer, and they're absorbed slower than other nutrients. So if you eat a really fatty meal, the duodenum is going to need more time to deal with it. Okay, so it's going to slow down your gastric emptying. The other thing is how acidic the chyme is. The duodenum has to neutralize the chyme. So if the chyme is very acidic, you're going to have to secrete more sodium bicarbonate, which comes from the pancreas and gets dumped into the duodenum to neutralize that acidity. So again, he doesn't want any more chyme until he can deal with that. So he'll slow down the contractions in the stomach. How hypertonic it is. If it's more hypertonic, it has more solutes. So it's going to take longer to absorb all those solutes. And then distension, the volume that he receives. He needs to deal with that volume before he receives more. So these are the things that will decrease gastric emptying. Now I told you that gastric emptying is going to be controlled by neural and hormonal mechanisms. And there's intrinsic, so these are your extrinsic controls. There's a reflex called enterogastric, and you should become familiar with these terms. Entero always refers to the small intestines. Gastric refers to the stomach, and then there's colon or colic, which refers to the large intestines. So enterogastric is a reflex that occurs between the small intestines and the stomach. So when the duodenum stretches because chyme is being put into it, that will be the stimulus to trigger the enterogastric reflex, which causes the pyloric sphincter to close. So some chyme gets put into the duodenum, it triggers stretch receptors, and that will trigger the nervous system to close the pyloric sphincter so that the duodenum doesn't just keep filling up. Then there's some hormones involved here too that affect gastric motility. You just learned that when chyme gets put into the duodenum will release cholecystokinin and secretin. And I told you cholecystokinin and secretin will go and inhibit gastric secretions. Well, it also inhibits gastric motility. So cholecystokinin and secretin are going to be released from the duodenum, and it's going to slow down the stomach both in secretions and motility. So here's a chart that shows that. You put fatty, hypertonic, acidic chyme into the duodenum. That's going to trigger stretch receptors. And there's a short reflex through the enteric nervous system that will close the pyloric sphincter. And then there's also through a long reflex, it will go through the central nervous system, increase sympathetic activity, decrease parasympathetic activity to inhibit the motility of the stomach. And then I told you about the endocrine cells in the duodenum. They're called enteroendocrine cells. Remember, entero refers to small intestines. And they're going to secrete what they call enterogastorones. So these are hormones that are secreted from the small intestines. So we talked about secretin, CCK, and we talked about gastroinhibiting protein. They're going to inhibit the contractile force of the stomach, therefore decrease its emptying rate.